Hello everyone, my name is Joanna Przybył. I'm at McGill University in Montreal, Canada. And thank you very much for having me. It's always a pleasure to present here. And today you will hear about our multi-omic study on desmoid tumors that revealed intra and inter-tumor heterogeneity. So as we all know, predicting recurrence of desmoid tumors is very challenging. It has been proposed that, for example, S45F mutation may be associated with higher risk of recurrence, as demonstrated in the studies of Dr. Alex Lazar, but today Dr. Schwartz actually showed some other contradictory studies, so this marker is still not used in routine clinical practice, and there have been also other clinical characteristics of the tumors or of the patients proposed to help predict recurrence of desmoid tumors, but none of them were actually universally validated and applied in routine clinical practice. So to help resolve this challenge, we performed a transcriptomic study where we profiled 20 patients. We profiled primary tumors from those patients, and 13 of those patients later, after resection of primary tumor, developed recurrence, and 7 of those patients did not develop recurrence. And we wanted to see what is different about those primary tumors that later behave differently. And because some of the primary tumors of those patients were quite large, we sampled actually multiple regions of those primary tumors. So from 6 patients, we performed multi-region analysis, and we performed transcriptome-wide RNA-seq. And in this study, we identified 14 genes that were differentially expressed between primary tumors that later developed recurrence compared to those that did not recur. And interestingly, when we plotted the normalized expression values of those 14 genes, we noticed that in some of the patients from whom we sampled multiple regions of their primary tumors, the expression of those genes was quite variable. So you can see that especially in the second patient, for example, plotted in this heat map, or in the last patient in this heat map. From those patients, we had 2 different samples, and you can see high variability of expression of some of those genes. So we became interested in looking more into this. We are actually not the first group who tried to develop a transcriptomic prognostic signature for prediction of recurrence in desmoid tumors. There are previous studies, one published in 2015 by the group of Dr. Frédéric Chibon, that identified a 36-gene transcriptomic signature that may be predictive of desmoid tumor recurrence. And also there has been a more recent study that identified a signature composed of 3 genes that may be useful in predicting recurrence of desmoid tumors. So we plotted those genes in our data set for validation and also to investigate whether we see similar degree of variability or not. And actually, as you can see, there was quite high degree of variability of expression of those genes, both within individual tumors and also across the 2 groups of patients that recurred or did not recur. So this was surprising, but also similar variability of expression of transcriptomic prognostic signatures has been previously reported in other types of cancer, in breast cancer, colon cancer, lung cancer. So we were not the first to observe this kind of variability. But this prompted us to investigate more in depth the potential heterogeneity within desmoid tumors. And for that, we focused on 3 patients from whom we sampled multiple different regions of their primary and recurrent tumors. So from patient 1, we analyzed multiple regions of the primary tumor and a recurrence that developed only 1 year after resection of the primary tumor. And we also analyzed multiple samples from patients who had 3 separate recurrences. And we performed whole exome sequencing, we performed the DNA copy number analysis using SNP arrays, we profiled DNA methylation using EPIC arrays, and we performed RNA sequencing. And today, for the sake of time, I'm going to present detailed data only from 1 patient, but I have extra slides from other patients as well, if we'll see how the discussion goes. So in the first patient that I will present to you today, this patient had a primary tumor of 19 centimeters, and we sampled 6 different regions of this tumor. And 1 year later, this patient had recurrence, and we sampled 3 different regions of this recurrent tumor. And importantly, this patient did not receive any adjuvant treatment after resection of primary tumor. And in all of the specimens sampled from this patient, we detected T41A mutation, but also interestingly, 2 regions of the recurrent tumor had a concurrent S45P mutation that here you can see we confirmed by Sanger sequencing, and it was also detected in exome sequencing data. Exome sequencing data also allowed us to do clonality analysis, and this analysis also identified some subclonal mutations, both within the primary tumor and the recurrent tumor of this patient. When it comes to DNA methylation profiles, we noticed that the primary tumor and the recurrent tumor had quite distinct profiles, but those profiles were uniform within individual tumor mass. So all of the samples that we obtained from the primary tumor had very similar profiles, and also all of the samples within the recurrent tumor had similar profiles. But when it comes to gene expression profiles, you can see more variability. So as you remember from the primary tumor, we sampled 6 different regions, and as you can see, one of the regions of the primary tumor had this cluster of highly expressed genes. You can see the sample here in the middle of the heat map, and some of those genes included, for example, pathways related to cellular metabolism. So this may indicate some differences when it comes to tumor metabolism. And in the recurrent tumor, you can see also that one region of the recurrent tumor had this cluster of highly expressed genes. And interestingly, those genes included some of the genes that belong to the Notch pathway, for example, Notch2. So I think it's an interesting observation, especially in desmoid tumors. So in summary, in this study, we demonstrated a rather unexpected degree of intra and intertumor heterogeneity of desmoid tumors, and this study shows that developing transcriptomic prognostic signatures may be rather challenging for desmoid tumors, and also that the analysis of a single tumor biopsy may be not representative of the full spectrum of molecular alterations that are present in desmoid tumors. And in addition to that, I think it's a very interesting observation that recurrent tumors actually show many different molecular alterations compared to the primary tumors. So I think it's an important consideration for drug development or therapeutic decisions for desmoid tumors. So I would like to thank the members of my lab who were involved in that study. I would like to thank collaborators from Stanford University, especially Matt van der Rijn, and collaborators from University of Toronto and Vanderbilt University. And I would like to thank Desmoid Tumor Research Foundation for their ongoing support through many, many years. And I would like to thank you for your attention, and I'm happy to answer any questions.
Great presentation. I wanted to ask you, well, this intratumoral heterogeneity that you're seeing, is this from multiple different clones, or are you seeing evolution of the same process but at different stages? In other words, if you think of this, say, similar to wound healing, right? Mm-hmm. And that there are different areas in the tumor, that they're all going through the same process, but at different stages and time. Mm-hmm, mm-hmm. Does the data support that? Or does it show that there is distinct, separate sort of seeds that are driving completely different stories here?
I think rather the latter, that in those different regions of the tumors, we rather see enrichment of different genes that belong to different pathways. So I think it's rather that, in the way I interpret this, is that some regions of the desmoid tumors maybe may temporarily show activation of certain pathways, like for example related to metabolic reprogramming or Notch pathway and so on. So I think it's rather that.
No, but say for example you're going from A to Z, right? It's going, and in this journey going from A to Z, there's going to be changes, right? But it depends when you're taking those pictures in the journey, and it could all be just part of the same continuum.
It may be. I just don't. I think based on the current data, we're not really. It's a very interesting point, but I don't think we are able to answer that, at least based on the data that we generated. But yes.
I have always question to myself that after the recurrence, some tumors growing up and growing up and growing up after the recurrence, but some tumors not. Recurrence and stable. So did you do the subgroup analysis of the after recurrence, stable and PD, the two subtypes, is there some difference of the RNA sequence? That is my question.
Yeah, I think definitely it would be interesting to look at that. Here, again, I think the cohort that we analyzed will not necessarily allow to answer this question. These are only 3 patients, and also each consecutive recurrence was actually surgically removed, so we don't have really this kind of, it's an interesting question, but I think, again, we don't have the data now to answer it.
Okay, so Dr. Kosaka's paper is that based on the most of samples from my university, so we couldn't do that kind of analysis at the time. So I'm very interested, curious about that. You have the recurrent tumors samples, so I hope you have more data from our Kosaka's paper. So.
We do.
That is my question, the reason.
So yes, I mean, your group has gracefully shared the data with us as well. It was used for other types of studies, but we have not done, so the signature that was published in that study, we did not attempt to repeat that analysis, right? That there were certain statistical methods and already a signature developed in the study. So what we show here is the signature developed in that study, but in our own data set. So it was kind of a different purpose, right? Just more for validation.
Okay. Thank you.
From the patients that you sampled, were any of them on therapy at progression? And did it differ, the therapy, in terms of the heterogeneity of the tumor?
Yes, so actually some of the patients, as you can see, they received chemotherapy, so like imatinib, for example, the different treatment regimens. So this, and we have seen a lot of heterogeneity in those kind of samples as well. And of course, we may suspect that maybe some of that heterogeneity may be related to treatment effects,
That's right? That was my question. Yeah. Is there selective pressure in the setting of a drug therapy to tumors evolve in the presence of a drug?
Yes, yeah, it definitely may be. So in this first patient that I presented here that did not receive any adjuvant therapy, actually compared to other patients, we observed a lesser degree of heterogeneity, I would say. In other patients, we see much more heterogeneity when it comes to transcriptomic profiles, DNA copy number profiles. Here are some examples, like, for example, there's a patient here in panel C where we can see a lot of DNA copy number alterations that are accumulated in some regions of the tumor. So definitely we cannot exclude that as well.
I have a question regarding the sampling. So multiple samples, spatially separated within the patient, within the tumor. When we look at tumors on imaging, there's going to be a lot of heterogeneity, and I wonder if there's been any effort to correlate the imaging location.
It would be definitely worthwhile, but we were not able to do that, to obtain this kind of information. So unfortunately, not. But yes, the signal can be heterogeneous as well, right?
Yeah.
That would be, we were discussing with some collaborators, it would be very interesting follow-up study, actually.
May I ask a sorry. You know, I'm kind of struggling with, I mean, all great presentations, but I'm, you know, Mushri, Christabel, your work. You know, in the clinic, we have drugs like sorafenib, which inhibits VEGF, PDGF, and then we have the gamma secretase inhibitors, now the beta-catenin inhibitor, possibly CD63 inhibitor. Is there a sort of a unifying theory here on why all these disparate drugs could work, or why are they working? Some say not, some say gamma secretase. Any sense, how do you make sense? Is there a unifying theory for why all these very different drugs are working?
From my point of view, I think we would need to analyze it in a more systematic way to have appropriate cohorts treated the same way and perform molecular profiling on this type of cohorts. Like here, what we look at are individual patients rather treated with different regimens. Even in this study, each patient received different kind of treatment and had different trajectories. So more systematic studies definitely would help us answer that.
If I may add, I think one of the problems is, you know, like let's say you treat a cohort of patients and you see 30% respond. Would this cohort been the same 30% that would have responded to also sorafenib or to another drug? Or is it always there's 30% that would have benefited from drug X and another 30% would have benefited from drug Y? I think just, at least from my perspective, I feel like desmoid tumors is a disease where it's threading a needle to happen. And so there might be multiple ways of trying to target it. And they're not necessarily, you know, the one unified pathway that we should go after, but there are multiple strategies. And at least from what we're seeing, it's probably different strategies for different patients. And I know it's like, you know, a super generic thing to say, you know, one size doesn't fit all, everybody says that, but I think that there is some truth to that.
All right, thank you very much.